morning students. Today I am going to explain about the development of female gametophyte. I hope you all understand my previous videos. So in last video I had explained you and there was types of the ovule, structure of the ovule I had explained you. Today I will explain you what is a macrosporogenesis and what is the development of female gametophyte. I am going to explain today. Okay. So if megasporogenesis, we have seen the microsporogenesis. Microsporogenesis is the formation of microspore from the microspore mother cell. Likewise, here also the megasporogenesis takes place. In the microspore mother cell will give rise to the microspore. Microspore is the first cell for the male gametophyte. First cell for the production of male gametophyte. Likewise, megasporogenesis is nothing but in this the process of development of megaspore from a megaspore mother cell is called megasporogenesis. So here the megasporogenesis process, the megaspore is formed from the megaspore mother cell is called the megasporogenesis. So you have seen in the male gametophyte in the microsporogenesis, that is microsporogenesis is initiated with archisporium cells only. Archisporium cells is nothing but there is an homogeneous mass of hypodermal homogeneous mass of cells is sequentially developing as a archisporium cells. This archisporium, some archisporium cells is directly act as the mother of the megaspore mother cell. Some will be again undergoing a transverse division. Through the transverse division, the outer primary parietal cells and inner sporogenous cells takes place. Okay. So, this inner sporogenous cells or the central sporogenous cells is act as the megaspore mother cells. So, this megaspore mother cells is give rise to the megaspore. Each megaspore mother cell will give rise to you. Yes, each megaspore will give rise to you. Each megaspore mother cell will give to you, that is 4 haploid megaspores. 4 haploid megaspore meaning is it's all the 4 cells. This one, yes, 1, 2, 3, 4. These all are the 4 megaspores. That is, in this last one is a functional megaspore. Why it is called functional megaspore? You may ask the question. Functional megaspore means it is going to be ready for development of female gametophyte, so that it is called functional megaspore. Remaining all these 3 megaspore going to get degenerate. So this is called functional megaspore. Okay. So the megaspore mother cells is undergoing a meiosis process. So that this is divided into 2. For you this meiosis 1, meiosis 2 no needed. For you only you can write it by the meiosis process. The megaspore, MMC means megaspore mother cells is producing the 4 haploid megaspores, in which the chalazal end and at the chalazal end, this is an chalazal end and this is a micropylar end. You consider like this is a chalazal end and micropylar end. What is a chalazal end? What is a micropylar end? I had explained you in the last video of the structure of the ovule. In the chalazal end of the functional megaspores, it is a functional one. This is going to give rise the female gametophyte. Remaining all 3 haploid megaspore is going to be the degenerate. Okay. So, if the 1 megaspore is giving rise to 1 female gametophyte is called monomeric embryo sac. If the 2 in the 4 megaspore, the 2 is involved and 2 is going to be giving the megaspore is giving the 2 is going to giving the embryo sac formation is called bisporic embryo sac. If the all also involved in the formation or the development of female gametophyte is called that is called the tetrameric embryo sac formation. Okay, so most of the 70% of the angiosperm only producing monosporic embryo sac from the one single functional megaspore. Okay, example polygonum type. If you are seeing the megagametogenesis or the formation of female gametophyte, nothing but there is in the mitosis 1, mitosis 2, mitosis 3, and these 3 mitosis 3 steps will be going to take. Okay, so the functional megaspore which is undergoing a mitosis process, 3 mitosis process going to take place. In the mitosis 1, this one cell is undergoing a nuclear division, not cytoplasmic division. If there is a cytoplasmic division, it is producing the 2 types of cells. But here only one cells, but it producing 2 nuclei through the mitosis 1 process. If you are taking a meiosis, this megaspore mother cell is a 2N, that is diploid one. But when it is producing a functional megaspore, it is producing N, haploid megaspores. So the diploid megaspore mother cell producing haploid megaspore in 4. Okay, so when the one cell or the nuclei which is undergoing a mitosis process, it does not change their ploidy number or the chromosomal number. What is a ploidy number or the chromosomal number? You may ask the question. Ploidy or the chromosomal number is nothing but the one nucleus is divided into 2 here. So this nuclei consists of 1 chromosome. This nuclei consists of 1. So there is in the 1 chromosome, that is N chromosome will be here. So it is called a haploid. When it is undergoing a mitosis 1, 2, 3, all those things, all the cells will produce as haploid type of megaspore only, no diploid here. So mitosis process does not change their chromosome number. Okay, so in the mitosis 1, the nucleus 1 is undergoing a mitosis process and produces 2 nuclei. This is a 2 nucleated haploid in nature in one cell. It is a 2 nucleated cell or the binucleated cell, you can say. And same again, it is undergoing a mitosis process. It is producing these 2 cells again, these 2 nuclei again divided into 4 cells. The 4 nuclei, the one is again divided into 2 nuclei. This one again divided into 2 nuclei. So it is a 4 nucleated stage or the tetranucleated stage of the megaspore. Again it is undergoing a mitosis. These nuclei also are haploid in the nature. If you are going the mitosis 3, in this mitosis 3, this one, these 2 undergoing a 4 nuclei, these 2 undergoing a 4 nuclei. So in this the 8 nucleated stage. This is called 8 nucleated stage. Okay. After the 3rd mitosis process, there is the wall formation takes place. That is cytoplasmic division only takes place after the 3 mitosis division only takes place. Okay. So in this, so if you see this, this is one cell. If you see this, this is one cell. So the wall formation takes place. So this is 1, 2, 3, 4, 5, 6. 6 cells are there. In 6 cells consisting 6 cells forming the cytoplasmic wall formation, and in between that you can see some 2 nuclei. So how it is separated, you may ask the question. So suppose you are considered this is a micropylar end of the ovule, this is a chalaza end of the ovule. The micropylar end will be having the 2 synergids. On the either side, left and right, you can appreciate 2 cells. That 2 cells is called synergids. The middle cell is called, that is female gametophyte or our embryo sac. And in below you see these 2 are there, these 3 are the antipodal cells. What cells? Antipodal cells. Only this female gametophyte, this embryo sac or the synergids involved in the process of the fertilization. These are all the antipodal cells or the simply cells. And if you see 2 nuclei, that is called a polar nucleus. This is 2 nucleus. These 2 nucleus, just before the fertilization, it is fused to form a secondary nucleus. Just before the fertilization, these 2 nucleus are fused and form the fertilization. All the nuclei will be having cell and these 2 nuclei will be having central cell. So 1, 2, 3, 4, 5, 6, 7. So it is a 7-celled 8 nucleated structure. You can count the nuclei will be 8, but you can count the cell, it will be in the 7. So it is a 7 cells and 8 nucleated stage you can find in the mature female gametophyte. Okay. If you see this diagram, now it will be explained you well. Okay, the embryo sac of the mother cell, that is this is a megaspore mother cell which is diploid in the nature. Okay, which is undergoing the meiosis process. Here they are not mentioning here meiosis. It is a meiosis process only. In the meiosis process, 4 haploid megaspores are produced. In this, the 3 are degenerated. The inner cell grows, that is the functional oneness in the chalazal end. It is undergoing the megaspore embryo sac, that is this one cell, and these for the 4 cell or the haploid in the nature. So it is growth is nourished by the nucleus. So it's undergoing a mitosis 1. So it is where it is going? It is going near to the micropylar end. Then again it is undergoing a mitosis process, it producing 4. Then it is undergoing a mitosis process, it producing 8. If you see this, these 3 at the one pole end, that is these 3 in the one pole end. This will be the yes, this will be the this is the chalazal end, and this is the where is the synergids is present and embryo sac is present, that end is your micropylar end. Where is the antipodal cells is present, that end is your this thing, that is your chalazal end. These are antipodal cell. But what is called egg apparatus means these 3 cells are called egg apparatus. The middle one is called ovum. There is a 2 nucleus there, that is called a polar nuclei. There are 2 nucleus there, that is called a polar nuclei. So 1 nuclei and 2 nuclei. These 2 nuclei fused just before the fertilization is called 2 nuclei. Okay. Then the nuclear fission takes place. Okay, so this fused, and now this cell, this female gametophyte is ready for the fertilization. Okay. Okay. Now you could have understand what is a megasporogenesis, and hypodermal cell in the nucellus is developing as the archisporium. Archisporium sometimes is directly act as the megaspore mother cell. Sometimes it undergoing a transverse division to produce as the outer parietal cell, and centrally primary sporogenesis. This primary sporogenesis is undergoing a megaspore mother cell. Megaspore mother cell undergoing meiosis process producing 4 haploid megaspores. Based on the number of megaspores, this is developed into embryo sac, monosporic, bisporic and tetrasporic. So monosporic and tetrasporic, this is if you see this thing. So if the 4 cells also involved in the mitosis process, it producing a 4 female gametophyte is called a tetrasporic. If the 2 is involved in the mitosis process and producing 2 female gametophytes, that is called the bisporic. And if the only one functional megaspore involved in the formation of the female gametophyte, that is monosporic embryo sac. The megaspore arranged in the linear tetrad. That only I had shown you in this diagram. It is arranged in the linear tetrad. This picture they are telling here. And if you see the megaspore, the usually chalazal one is a functional and other 3 are the degenerates. Okay, if you see this is one is chalazal end. If you see the inverted ovule, this is a chalazal end. Chalazal end having one megaspore is a functional. Remaining all are degenerated. So next I am coming to here. The functional megaspore forms the female gametophyte embryo sac. Monosporic development is example. Monosporic development example of the plant is polygonum plant. Bisporic example is allium. Tetrasporic example is peperomia. And ovule generally has a single embryo sac. If you see the ovule has a single embryo sac. Development of monosporic embryo sac, polygonum type. Next, the functional megaspore, first cell of the embryo sac or the female gametophyte. The functional megaspore is first cell of the it is a first cell of the female gametophyte, like we saw in the microspore. That is, the microspore is the first cell for the formation of female gametophyte. The megaspore elongates along the micropylar and chalazal axis. The nucleus undergoes a mitotic division. The nucleus undergoes a mitotic division. Wall formation does not follow the nuclear division. Yes. Nucleus undergo only the mitotic division, wall formation does not take place. You see in this diagram, only the nucleus division takes place. At one particular stage, only the cytoplasmic wall is formation. Till that, there is only the nuclear division. So, in the mitosis, there is no cell wall formation. Only the nucleatic divisions and nucleus also haploid in the nature. The ploidy number or the chromosomal number will not change in the mitosis process. And if you see the structure of the beautiful structure of the female gametophyte, this is antipodal cells in the chalaza end, and the micropylar end will be having a synergids with the egg or the female gametophyte. So this egg along with the synergids is called as an egg apparatus, which is given in your textbook, and these are all the polar nuclei, that is the polar nuclei in the center cell. So this is one cell, this is one cell, this is one cell. So this is totally is called a 7-celled 8-nucleated structure. So the 4 nuclei at the micropylar end is 1, 2, 3. 3 will be the micropylar end of the 3 nucleus is organized into a egg apparatus. Okay, the 4th one is left in the central one. So the polar nuclei they are telling in the, it is a central one, and 3 nuclei of the central one, it is a upper polar nucleus and lower polar nucleus. These 2 nucleus again it is divided into upper polar nucleus and lower polar nucleus. The upper polar nucleus, this is 1. Okay, and the polar nucleus, lower polar nucleus is just near to the chalaza end of the antipodal cells. The 2 polar nuclei fuse to form a secondary nucleus just before the fertilization. I had told you already. The egg apparatus is made up of central egg cells and 2 synergid cells. What is the role of synergids? You can ask me the question. Synergids going to secrete some chemotropic, some chemical substances, that too it is attracting the pollen tube. And filiform apparatus of the synergids, it helps in the absorption and conduction of the nutrients from the nucellus to the embryo sac. So embryo sac, which is growing in the ovule, it should get the nutrited, that is done by the filiform apparatus. Okay, and it also guide the pollen tube. So the role of the filiform apparatus, it's giving the nutrition to the embryo sac as well as it is guide the pollen tube. Okay, it is a 7-celled and 8-nucleated embryo sac. Don't forget this term. 1, 2, 3, 4, 5, 6, 7, okay, 3, 4, 5, 6, 7, 8. 8 nucleated 7-celled stages can take place in the matured female gametophyte. And also I told you under the mitosis process that the ploidy number will not change. There is a no cytoplasmic division. Only the nuclear division will be takes place. Okay. Next. So this is nothing but structure of female gametophyte. It is also called a embryo sac. In angiosperm, it is mostly develops as a 7-celled 8-nucleated structure. It consists of 3 antipodal cells, 1 central cells, 2 synergid cells, and 1 egg cell mostly. So that is they have given micropylar end, chalaza end, antipodal cells, polar nuclei with the 2 nuclei. Okay, this is a central cell. Then this is just nothing but megasporogenesis. What is megasporogenesis? What is microsporogenesis? Difference between I have given you. And what is a male gametophyte and female gametophyte? This is they may ask you in the 3-mark questions. If you are going in the AIMS exam, in the NEET exam, they will be asking mainly, the most frequently they are asked what is the ploidy or the chromosomal number of the mitosis of the female gametophyte, they will ask. So in the mitosis does not change their ploidy number or the chromosome number. They always will be the same in their chromosomal number, and it is always in the haploid in the nature. It is follows only nucleated structure. It does not go for the any cytoplasmic division. Cytoplasmic wall formation only takes place after the 3 mitosis process. Hope you all understand this video. Thank you.